Welcome back friends, how are you all? Here is the Gene Island School and here we will learn about genetic engineering, biotechnology and molecular biology. Share with your friends comments about your topic, what you want to know. Today our topic is one type of gene therapy that is called antisense gene therapy. At first we have to know what is gene therapy. Gene therapy is the treatment of any disease with the gene. With the treatment of gene, we can cure from a disease. That is called gene therapy. Gene therapy may be replacing a disease gene. We can replace a disease gene with a good gene, or we can knocking out. Knocking out means we can stop a gene that is producing non-functional protein that is causing disease. We can knocking out, or that we can switch off. We can switch off the gene that is producing non-functional protein. There is one type of gene therapy. And here is a new type of gene we can produce for the betterment of the body cell. That is called another type of gene therapy. So the moral is that gene therapy is the treatment of any disease with the treatment of gene. Normally we treatment our disease by medicine. How do you know that? Medicine affect our cell membrane, etc. and can relieve us from any type of disease. But in the gene therapy will not. In the gene therapy, the treatment is occurred not among the membrane, cell membrane. That is inside the nucleus. That means inside the nucleus, DNA and the gene. That is the difference between the conventional therapy and the gene therapy. Here we comes our main topic. That is antisense gene therapy. That is sense. Antisense. That means antisense therapy. That is type of treatment by which the genetic sequence of a particular gene is known to be causative of a particular disease. When or non-functional, we know that we know in our body a gene is producing a non-functional protein. This protein is affecting our body. We can stop this. We can stop these proteins translating. That is with the oligonucleotide. When we attach, here is the figure, we'll really see that. That is the DNA. We know from transcription we get a mRNA, and if we attach the antisense DNA oligonucleotide with the mRNA will not be capable of translation. When that is attached to a single strand of mRNA, that mRNA then turned into a double stranded. We know that if the single stranded mRNA is not normally occurred or is not normally worked, then it will not produce any protein, and we know the protein is the main factor of characteristics of our body. Here you see that if it is disease causing DNA, if we attach with oligonucleotide, then it will not produce any protein. That is the main mechanism of antisense gene therapy. Here is the reading. Antisense therapy is a form of treatment for genetic disorder or infection. When the genetic sequence of a particular gene is known to be causative of a particular disease, it is possible to synthesize a strand of nucleic acid that is bind to the messenger RNA. Here, that is the oligonucleotide bind to the messenger RNA, and that is inhibiting the protein translation. And what is the oligonucleotide? Oligonucleotides are a chemical synthesized that use phosphoramidase, and oligonucleotides use normally 3 to 5 terminals, because this mRNA is 5 to 3 terminals. So the antisense DNA oligonucleotide normally will be 3 to 5 terminals. And here is the oligonucleotides are the molecules made of synthetic genetic material. That is made with synthetic genetic material. And what is the advantage of oligonucleotides? The main advantage of oligonucleotides are that is manufactured quickly within a week. So we can get a oligonucleotide within a week. So that is so much easy, and that is easily measured. We can measure a oligonucleotide therapy. This therapy we can easily measure. What is the oligonucleotide is working in the experiment. Another advantage of the potential to produce longer lasting response. We know that in conventional therapy, longer lasting response are normally not found, but if we use gene therapy for any disease treatment, then it will be longer lasting and maybe lifetime, protect our body from the non-functional protein producing or disease functional protein producing. So that is so much effective. Here is the another advantage: potential for enhanced binding affinity to target. It is a potentiality. That means it has a binding affinity to the target. Oligonucleotide has a binding affinity. We know that oligonucleotide bind with the mRNA. That is, if it has easy binding capability, then it is so much easy for the response to get result. That is the oligonucleotide, and that is the target, and here is the target binding, and here is the another advantage. In the next step, what is challenges? We know that antisense gene therapy is a modern technology. Antisense gene therapy is genetical technology that may have some challenges. One major challenge is that antisense therapy, it is difficult to enter into our body. Our body is affected. It is not so much easy. We can easily inject this into our body, oligonucleotide. That is an experimental way. Here is the example that delivery of the treatment of the brain. If our brain is attacked, our brain is diseased, it is not so easy. Like for used to disease like HD disease, when it affected by our brain, affected to our brain, especially challenging because it must cross the blood-brain barrier. Blood-brain barrier is so much sensitive, and that may a great challenge for the antisense gene therapy. Another type of challenge is that toxicity. We know if our body get a new type of, here you see that our body is getting a new type of oligonucleotide, antisense DNA oligonucleotide. Body will think that is an antigen, and body can effect to this by WBC, and that may cause toxicity. There is a simple way. So another challenge is that toxic effect. Although antisense technology is engineered to be very specific, it can still cause unintended damage. Still, it can produce unintended damage to our body. But experiments are working, and scientists are developing day by day. So that's the main details about the antisense gene therapy. Here is the full review. What is gene therapy? Gene therapy is the treatment of our body with the gene, among the gene, inside the gene. That is gene therapy. Gene may be new gene may be introduced, some gene may be knocked out, or replace. We can replace the affected gene with a new gene. That is the main action of the gene therapy. And the antisense gene therapy, as said that here a DNA transcription, by transcription we get mRNA. But when mRNA is attached to an antisense DNA oligonucleotide, these will form a second stranded form and that are not capable of translation and capable of produce protein. That's all about antisense gene therapy. Friends, if you like this, share it with friends, comment, ask what you want to know. Thanks everyone and keep stay with Gene Island School.